My name is Anna Driscoll. I go to the University of Pittsburgh, and I am a bioinformatics major there, and I'm working in Dr. Jamie Walters' lab this summer. We can quantify sex-linked genes because males and females have different numbers of sex chromosomes, therefore they'll have different numbers of sex-linked genes. I hope to do this using statistics meant for quantifying RNA instead of DNA because RNA quantification requires significantly less data. So the weirdest thing that happened is we were trying to account for some transcripts that we didn't know where they came from, and I looked them up, and one of them matched the sequence of a fire ant, and given we're working with butterflies, that's not supposed to happen.
My name is Asha Navarro. I go to MCC Penn Valley in Kansas City, and this summer I'm working in KU at the museum in the herpetology division. This summer, my research was collecting external morphological data in order to test the hypothesis that genetically distinct populations of Anolis cysticus are worthy of elevation to full species. Now, I thought they were really cute. Once seeing them under the microscope, they seemed real, but not real. And it was just, I just got over it.
My name is Autumn Peterson, and I go to Virginia Tech in Blacksburg, Virginia. I am looking at the morphological evolution of Cupressaceae, and I'm looking at a new specimen from the Paleogene of North America and using that data to shed light on the evolution of Cupressaceae and different characteristics that diverge over time. Cupressaceae is a type of conifer. They are native almost everywhere, especially in the northern hemisphere. We do a process called the cellulose acetate peel technique, where we grind and etch the fossil and place a peel on top. That way we get small microns of rock layers, and there we're allowed to easily peel off each layer and see what comes out and what doesn't.
My name is Armelis Romero Ponce. I came from the University of Puerto Rico at Maya West campus, and I'm in my third year of biology. We are measuring the phosphorus that becomes available to trees in contrasting forests over a year of dead leaves decay. And I do this by quantifying the phosphorus that is in rainforest that has been leached through each forest from the leaves. We know how much phosphorus the trees need for an annual demand, but we don't know how much they get from organic matter decay. And since they can obtain this from organic matter or from the bedrock, it's important to know how much do they get from organic matter decay, because the rocks are not going to be here for a long time, and if they get most of their nutrients from the bedrock, that means that the forest will be in danger maybe in a couple of thousands of years.
My name is Ben Papadopoulos. I go to the University of Puget Sound, and I am a rising senior, and I am studying biology with a bit of a focus on evolutionary genetics. So my project focuses on exploring how the likelihood of increased drought with climate change will affect the relationship between soil microbial communities and the plants they associate with in preserved and disturbed soil. Well, as climate change makes precipitation events less frequent and then more severe when they do occur, it's important for us to know how that'll affect soils and the ways that our plants grow so we can sustain the necessary land use that we have. So hopefully through this we'll be able to know what conditions will facilitate responsiveness and resistance into the future.
So my name is Carrie Spanton, and I go to Metropolitan Community College, Penn Valley campus in Kansas City, and I'm studying ecology. There are microbial communities in the soil that both consume and produce methane, which is a critical greenhouse gas that affects our climate. We studied how land use management and climate affect these microbial communities.
We found that native prairie land had a higher methane oxidation rate than agricultural land, which is important because how we use our land has a lasting impact on microbial communities.
I'm Hannah Day. I go to Benedictine College and I am a biology major. This summer I am studying the effects of fire severity on the interaction between soil nutrients and microbial communities and their impact on the growth of a fire-tolerant and non-fire tolerant species of pine. Understanding how soil nutrients and microbial communities interact after a fire will help land managers of savanna ecosystems to make more well-informed decisions on how often to burn, and this will help preserve these endangered ecosystems.
Hello, I'm Griffin. I'm a senior at the Evergreen State College in Olympia, Washington, and this summer in the MORT lab we use genetic, morphological, and biogeographical data to estimate mating systems and infer divergence in a species of North Atlantic flowering composite called Tulpis succulenta. This study is important because it gives us insight into how these plants are colonizing islands, which is important to understand in the more general case. Also, knowing how these plants mate in the wild and what they're doing is really important for developing a comprehensive conservation strategy. But above all else, the pursuit of knowledge is self-justified.
I'm Hannah Swan, and I go to school at Hamilton College, which is in upstate New York, and I'm actually a math major, but this summer I'm working in Dr. Gleason's Evolutionary Biology lab. So this summer I'm working with a species of fruit fly, which is Drosophila sturtevante, and I'm looking at their courtship behavior. And one of the behaviors that they exhibit when they're trying to mate is they vibrate their wings and sing a song, and we're trying to characterize the attractive components of their courtship song. I have never worked in behavior before, and I learned that one of the best mechanisms for getting them to sing a lot is putting a beheaded or dead female in with the male, and that was the trial that I got the most song on.
Hi, my name is Isabel Penn, and I work in Dr. Jensen's lab studying the tapeworms of sharks and rays. For my project, I am using light microscopy, scanning electron microscopy, and histological techniques to redescribe Mysocephalus nari-nari, which is a tapeworm found in the spotted eagle ray, and it hasn't been reported since 1906. There's this thing called microtriches on the, like if you look on these, it's like kind of fuzzy. Here, I have a picture of it. There are these things called microtriches that are on the surface of all of the tegument of the cestodes, and we are the first ones to look at the microtriches for Mysocephalus nari-nari and for the entire genus of Mysocephalus as well. So the first microtriche data that has ever been procured for Mysocephalus.
So hi, my name is Layla, and I am going to be a sophomore at Virginia Tech. I am studying biological systems engineering. So here we are investigating nosema, which is a gut parasite that infects bumblebees, and we are trying to find if the samples that we have collected from the field have died because of nosema or not. Well, because the parasite we're looking for is a specific gut parasite, and if we don't look at the guts of the bumblebees, we'll never know if they got it or not. That's why we kill them, to find out what's killing them. And if you think about it, it's not bad, because if we don't kill them, they will die themselves, and no one will ever know what happened.
So my name is Maggie Flowers. I'm going to be a junior, and I'm working in the ecology field. My research is focused on an insect sampling method called pan-trapping. We want to know if pan-trap height above the ground, pan-trap diameter, and the floral density at fields in which pan-traps are set will influence the abundance and body size of our captured bees. So my research is important because there's currently a worldwide decline in bees, and without a standardized sampling method, ecologists will be unable to conduct comparative studies. Pan-trapping is just a sampling method, and small plastic bowls are filled with soapy water to trap and drown the insects, and they're usually painted yellow to attract pollinators.
My name is Megan Resurrection. I attend the University of Illinois at Urbana-Champaign. I currently major in actuarial science, and I have minors in creative writing, informatics, and mathematical statistics. My project was on the effects of breeding peak phenology in an avian malaria transmission model for Hawaiian birds and mosquitoes. We ran simulations with an SRM model to predict when the highest host infection rates are, so we can take conservation measures to protect these bird species from avian malaria. I think overall my experience this summer has been good. I've been able to gain research experience for the first time, and being able to work in a lab, modifying code and running it, and being able to write a paper and do all the things that a scientist actually does is good for me, and especially since I want to go to graduate school someday.
So I'm Rachel Elting. I go to Johnson County Community College, and I'll be transferring here next semester. I'm going into bioinformatics. So I identified sequence reads from postman butterflies, which most likely came from the W chromosome. And I did that by taking long reads from females and aligning a bunch of short reads from males and females to it. And if only short reads from the females align to the long read, then that long read most likely came from the W. So the long read is from a female, and if only the female short reads align to it, then that means that that long read is probably from the W chromosome, because the W chromosome is only found in females.
My name is Chelsea Espino. I study at Avila University, and I know this is a research program, but I'm actually pre-med. My project focuses on analyzing soil samples from 3 regions in Kansas. We want to see if the amount of precipitation and the type of land use affects the levels of nitrate and ammonium in the soils, in hopes of better understanding one of many processes within the critical zone. The critical zone is the area from the outer extent of vegetation, so like the leaves from a tree, all the way down to groundwater. And it's important because it forces scientists from different fields to come together and learn more about the systems that are crucial for human sustainability.
I should do this one. Hello, my name is Isabel, but no, I'm just kidding, I'm not gonna do that one.
Hi, I'm Griffin. I'm a senior at the Evergreen State College.
Statistics are useful for this. Oh, that wasn't good.
Let's do it one more time.
Why?
Were you grossed out when you started with it?
No, I wasn't. Well, a little bit. I was nervous of breaking them.
My junior mentor, great dude, but he definitely spilt beta-mercaptoethanol all over his beard and his shirt. It permeates your cell membranes and binds to your DNA. It's really toxic, carcinogen, but he powered through it, didn't even bother to change his clothes.
There's 14 seconds.
Yes! Ew, there's a hair on this wall.
But I maintain that the explosion in my professor's centrifuge, that was not our fault. That was about the Falcon tubes. Not my fault. Actually, yeah, maybe edit that one out. That's Jenny's not gonna want it.
Oh, dude, do you want to see butterfly?
Should I smell it, or do you think that'd be weird? No, it's too long.
It's fine. You're doing fine.
Ah!